Hello students. Today in this video we'll study about the next chapter of biology, that is heredity and evolution. Now, have you ever wondered why humans give birth to human child? Why a dog can give birth to only the puppies? And even if you sow the seed of a maize plant, it will produce a maize plant itself. Why it is so? How come the cell know which characteristics to develop? So that is what we'll try to understand in this chapter of heredity and evolution. That branch of science which deals with the study of heredity is known as genetics. And heredity is transmission of characteristics or genetic information from one generation to the other, from parents to offsprings, is known as heredity. And the mechanism by which the information is passed on from generation to generation is known as inheritance. Now, many scientists, they tried to study the mechanism of inheritance, but it was Gregor Johann Mendel who was able to study in detail about the laws and the mechanism of inheritance, and he could produce it to us. Now, let me first tell you, the characteristics or the information, they are stored in each and every cell. As you know, all the cells have nucleus, and nucleus contains thread-like structures which are called chromosomes. These chromosomes are made up of DNA. The unit of DNA are the genes. Now, what happens? The information is stored in the genes. When the parents produce the offsprings, the information is passed on through these genes. There are thousands of genes in the chromosomes, and each gene is responsible for our characteristics. For example, your height, the color of your skin, texture or color of your hair, color of your eyes, all these characteristics are determined by the genes which you inherit from your parents. This is all about inheritance. So Gregor Johann Mendel, he performed various experiments on pea plant. The pea plant, the garden pea. He used to stay in a monastery where he used to grow the pea plants. He also had knowledge of mathematics and statistics. So he applied his science and maths knowledge to analyze the results of these experiments. That is why he was successful and able to tell others how the inheritance is possible, how the information is passed on to the next generation. Before we move further, let me tell you, he chose pea plant. The scientific name is Pisum sativum. Why he chose pea plant? Firstly, pea plants are very easy to grow. Secondly, the pea plant, the flowers of the pea plant, they allow self-pollination. However, cross-pollination is also possible. So different breeds could be bred to produce new generation of pea plants. Thirdly, each plant gives rise to number of seeds. But the most important reason is the pea plants, they show contrasting characters, few contrasting characters. So it was easy to compare them. For example, some pea plants are tall while some are dwarf. Then some bear the violet color flower, the others have white color flowers. The seeds of the pea plant are either round or wrinkled. The color of the seeds is either the green or it is yellow. So thus such contrasting characters, it is easy to compare and study them. That is why Mendel chose pea plant for his experiments. Now, the first experiment which he performed was monohybrid cross. In monohybrid cross, there is a cross breeding between two plants considering only one pair of contrasting characters. Okay, so let us consider this monohybrid cross between a tall plant and a dwarf plant. Now each character is represented by a pair of genes. So a tall plant is represented as a pair of capital T. This is a pair of genes representing a tall plant. On the other side, he took another plant which was dwarf. For dwarf, we'll take a pair of genes which is represented by small letter t. Alright, so there is a cross breeding between tall and dwarf. As you know, what happens during gamete formation? In sexual reproduction, One male gamete, one female gamete fuse together to form a new cell, which is called zygote. Now during gamete formation, what happens? The chromosomes, they separate out. So gametes contain half the number of chromosomes. For instance, human beings, each cell contains 23 pairs of chromosomes, that is 46 total chromosomes. But the gametes will have only 23 chromosomes, half the number of chromosomes. So half from the male, half from the female, these chromosomes combine together to form a new individual. So in this case, when gametes are formed, each gamete will have capital T, alright, and in this dwarf plant will have the small t gametes. These are the gametes. Now during crossbreeding, what will you expect when when pollen grains of tall plant and let's say ovum of a dwarf plant, they are fertilized and a new plant is formed. You might expect that a medium-sized plant will be formed, but no. What Mendel observed was all four were tall plants. All right. So when this gamete fuses with the other gamete, it will be the new zygote will have capital and small t. If this fuses with the other gamete, again capital and small. But if there is a chance pairing of this gamete with this, it will again have capital and small, and the fourth case also it will have one capital, one small. This is called F1 progeny or the first generation. In the first generation, what was observed? That all the plants were tall. So that means although they had a gene from the dwarf, but this dwarfness could not express itself. So such genes which cannot express themselves in the presence of another gene are called recessive genes. And the other gene which can express itself even in the presence of recessive are called dominant gene. So that is why we have used capital T for a dominant gene tall and small letter t for the recessive gene dwarf. So it was observed that all the plants grown in the first generation all were tall, although it was expected that it might lead to a medium-sized plant. So all plants were tall. So that is why tallness is considered as a dominant gene, dwarfness is considered as a recessive gene in pea plants. Now, what Mendel did was he performed self-pollination with these plants. So when a gametes will be formed from this, what will happen? Half the gametes will have capital T, the other half will have small t. So each plant will lead to two types of gametes. Capital T and small t. Half the gametes will have capital T, other half will have small t. During self-pollination, what will happen? If this gamete will fuse with this one, it will give rise to capital T, capital T. But if this fuses with the other one, it will have one capital, one small. Now, let us consider this gamete fuses with this capital T, small t. But in fourth case, notice, this small and this small, they'll fuse together to form two small t's. Right? So in this case, this will be a tall plant. Why? Because it has both the genes of tallness. This plant will also be tall. Why? Because it has one dominant gene of tallness, one recessive gene of dwarfness. So recessive will not be able to express itself. So, but dominant, because of the presence of one dominant gene, this plant will also be tall. Next, this plant again will be tall because of presence of one dominant gene. But in this case, notice, both the genes are dwarf, that is small t. So this plant will be dwarf. So in this F2 generation, what was found? That there were 3 tall and 1 dwarf. Tall and dwarf plants were in the ratio of 3:1. This ratio of 3:1 is called monohybrid ratio. So just imagine, the first generation had all the tall plants, but the second generation has 3:1 ratio of the dwarf plants. So where were these dwarfness gene in the first generation? It could not express itself. So that is what Mendel suggested, that the dwarfness is a recessive gene which could not express itself in the F1, but it was able to express itself in the F2 generation because there were no dominant gene there. So that is this gave rise to first law of Mendel. Mendel suggested that during gamete formation, the chromosomes, the genes, they separate out, they segregate to form half-half to each cell. This is called law of segregation. During gamete formation, the genes, they segregate and only half of the gene enter into each gamete. And during sexual reproduction, two different genes combine together to form a new organism. And this ratio of 3:1 in second generation F2 progeny is called monohybrid ratio. Now let us move on to the next type of experiment, which is dihybrid ratio. Now, the second type of experiment performed by Mendel was dihybrid cross. We just discussed about monohybrid cross. This is dihybrid cross. In this, what happens? Two plants, there is cross breeding between them, considering two pairs of contrasting characters, right? So in monohybrid, we considered only one contrasting character, like tallness and dwarfness. In this, we'll take into consideration two characters. So during this crossbreeding, what was noticed, that came out as a law of independent assortment. During gamete formations, the genes, they separate out, that is law of segregation. Now one pair of genes for one character, they pair with another pair of genes of another character independently. That is second law of Mendel, law of independent assortment. Let us study how it happens. Now, for example, I have considered an example of round green seeds and another parent having wrinkled yellow seeds. Now, the seeds are either round or wrinkled. This is one character. Since round is a dominant gene, I will take capital R as a symbol. While wrinkled is a recessive gene, we will take as small r as the symbol. On the other side, another character is the color of the seeds, which is either green or yellow. Yellow is a dominant gene, so I have taken capital Y as the symbol, while green color is a recessive gene, so small y for this. So this plant has capital R and small y, while the other plant has small r and capital Y. Now, during gamete formation, the same types of gametes will be formed, one from R and another small y. So this gamete will have one gene for round and another for green color of the seed. While in this case, the gametes will have small r for the wrinkled character and for yellow, capital Y. So these are the gametes formed. Now during cross breeding between these two plants, what would happen? If this gamete fuses with this, what will you observe? Capital R and small r from here. Then capital Y and one small y from here. So, since there is one capital R, so the seed will be round. And one capital Y, the seed will be yellow. Right? Similarly, if this gamete fuses with the other gamete, if there is a chance fusion, it will again be R, R, Y and small y. Then this between these two gamete, again same thing. These two gametes. So you will notice in F1 progeny or the first generation, all the seeds are round and yellow. So that is why we considered round as the dominant gene. Similarly, yellow color as the dominant gene. While the wrinkled, the small r, is a recessive gene. It could not express itself in the first generation. Similarly, small y is for the green color of the seed. Even that could not express itself in the first generation. So all the plants bear seeds which are round and yellow in color. This is the F1 progeny. Now, these plants were allowed to have self-pollination. What types of genes will be formed? These are can pair with capital Y, capital R with capital Y, right? This is the first type of gamete. Or capital R with small y. This is the second type of gamete formed from the F1. Then the small r may pair with the capital Y. This is the third type of the gamete form. And lastly, small r with small y. So these are the 4 different types of gametes which were formed from the F1 generation. During self-pollination, either of these will fuse together to form the F2 generation or the second generation. Now, just try to do it yourself. If this fuses with this gamete, what will you observe? Capital R, small r. So it will be round. Capital Y, it will be yellow. So 4 types of, we'll just discuss how 4 types of the plants were observed. Although we took only 2 types of parents, but in the F2 generation, Mendel found 4 types of plants. First, round and yellow, then wrinkled yellow, then round green and wrinkled green. So 4 types of seeds were observed in the F2 generation. The ratio was 9:3:3:1 of these 4 plants. This ratio is called dihybrid ratio. 9 out of 16 plants, this total is 16. So 9 out of these 16 plants were round and yellow, that is both the dominant genes. 3 were round and green, 3 were wrinkled yellow, but only 1 was wrinkled and green in color, both the recessive genes. This one represents wrinkled green, both the recessive genes. Now, in the tabular form, I'll explain you how we found this ratio. Now, consider these 4 as the gametes of one parent and these as the gametes of the other parent, the male gamete and female gamete. Now, if this capital R, capital Y fuses with capital R, capital Y, we'll find this F2 progeny. This will be round and yellow. Again, this fuses with this gamete, capital R, capital Y, round yellow. So this way you can frame the 16 types of plants formed in the F2 generation. Out of these 16, you'll notice 9 of them have round yellow seeds. If there is even one single capital R, the seed will be round. Even if there is one capital Y, the seed will be yellow, right? Because they are dominant gene. They'll express themselves even in the presence of recessive gene. Now, this is the first type of plant, that is round and yellow. We found them to be 9 out of 16. The other type would be round and green. Let's see. This one is round and green. This one is again round and green. One capital R, so round, but both are recessive, so round and green. And the third type, if you can find out, round and green. This round for capital R, small y will represent green. So 3 of them are round and green. Then comes wrinkled and yellow. Okay, this one, wrinkled and yellow. Small r will represent wrinkled, capital Y will represent yellow. So wrinkled yellow, and this one again wrinkled yellow. So 3 of them will be wrinkled yellow. But wrinkled and green, both recessive, you will find there is only one, this one, wrinkled and green. So the ratio is 9:3:3:1. 4 types of plant in the ratio 9:3:3:1. This is called dihybrid ratio. From this, Mendel could tell us that during gamete formation and during fertilization, what happens? The pairs of the genes, they separate out and they form new pairs independently. Okay, so that is law of independent assortment. Now let us move on to the next topic. Students, the next topic under heredity is sex determination in humans. Now, whether the child will be a girl child or a boy child, this sex determination in human beings is genetically controlled. As you know, we have 23 pairs of chromosomes. Out of these 23 pairs, 22 chromosomes, 22 pairs of chromosomes are called autosomes. They control the normal functioning of the body, like the characteristics, the external features, or the other systems, digestive system, respiratory system. They are controlled by the other 22 pairs. But there is one pair of chromosomes which are called the sex chromosomes. They play a role in determining the sex of the newborn baby, the new offspring. Now, in males and the females, that is both the parents, 22 chromosomes, 22 pairs of chromosomes are identical, they are similar. But the 23rd pair is called the sex chromosome. It is different for males and different for females. In males, it is termed as XY chromosomes, but in females, both the chromosomes are identical, so there is X and X. So during gamete formation, they separate out, the pairs separate out to half the number. So in males, the gametes have, half of the gametes have X chromosome, while the other half have Y chromosome. But in female, when gametes are formed, the gametes are similar. All the gametes will have the X chromosome. When you make it into half, X and X. So all the egg cells, that is the female gamete, will have X chromosome, but the sperm cells, half the sperm cells will have X chromosome, the other half will have Y chromosomes. Now what happens during fertilization? One of these sperm cell will fuse with the egg cell. So if the X chromosome of the male fuses with this female gamete, the next zygote will have XX chromosome. This zygote will grow up to a girl child because it has XX chromosome. But if the sperm bearing Y chromosome fuses with the egg cell, it will have X and Y zygote. This zygote will grow up to be a boy child, right? So this is how the sex is determined for the new offspring in human beings. So that means whether you will be a boy or a girl, whether the new offspring will be a boy or a girl, it depends upon the genes which it has inherited from the father, from the male parent, not from the female. So it depends upon the sperm which fuses with the male gamete, with the female gamete. That is how the sex of a newborn baby is determined in a human being. I'll take another example of blood group. The blood group is controlled by three genes which are represented by the symbol IA, IB and IO. It is represented like this. So, out of these three, A and B, they are dominant gene, while this O1 is a recessive gene. So as you know, there are four blood groups, A, B, AB and O. So if a person has a blood group of A, it might be because of presence of this gene, right? Both are dominant, or it might be because of presence of a pair of gene like this. Since O is recessive, if a child got O from the father and A from the mother, it will have A blood group because O is recessive and it will not be able to express itself. But this A gene is dominant, so it will be able to express itself. Similarly, if a child has blood group B, that can be because of both the dominant genes, or it can be because of one dominant B and the other recessive O gene. If a child has blood group AB, this is because both the dominant genes are present, A as well as B. Both are able to express itself. I told you, both are dominant gene. So the child will have the blood group AB. But to have a blood group O, both the genes need to be recessive. Then only the child will have the blood group O. If this is present with a dominant gene, then it will not be able to express itself. Recessive genes can express themselves only if both the genes are similar. That is why only this child will have a blood group O. So, if you have a blood group A, that doesn't mean that you have both the dominant gene or both the parents need to have A. It can come from this combination also, right? Now, next question, how is equal genetic contribution of male and female parent ensured in the progeny? Okay, so in sexual reproduction, I told you that firstly gametes are formed. These are gametes which contain half the number of chromosomes. Then half chromosomes from male and half chromosomes from female, they fuse together and a recombination of genes occur. So in sexual reproduction, you can see the 50% of the chromosomes are from the male parent and the other 50 are from the female parent. So there is equal contribution of male and female in sexual reproduction. Next question, how do Mendel's experiments show that traits may be dominant or recessive? So if you have seen my video, I told you Mendel, he performed two kinds of experiment. First was the monohybrid and second was dihybrid. So how did he show that traits may be dominant or recessive? This was shown by monohybrid cross. So for that, Mendel took pea plant, okay, one was pure tall plant, another was pure dwarf plant, okay. Capital T refers to the tall plant, it's a pair of genes, a trait which will show tall plant, and this one is a dwarf plant which is shown by a small letter t. Now first of all, first step is gamete formation. So in this case, both the gametes will contain capital T. In this case, both the gametes will contain the smaller t. If the parent cell contain both the capital genes, I mean it is only symbolic although, then it is called a pure tall plant and this is called a pure dwarf plant. Okay, the size of the plant is smaller. So the cross breeding was done with anther of ovule of one and pollen grains of the other. Cross breeding was done. What was observed in first generation, all the plants were tall. None of the plant was dwarf. Why? How it happens? If you cross breed them, so it will be one tall, one capital T and one small t from each parent. Then this also can lead to, so all types of combination if you make, what you will notice is all of them have similar kind of chromosomes containing one capital T, one small t, one from the male parent, another from the female parent. So during F1 generation, all the plants were tall. In the second step, what was done that these parents were taken as the parent. Okay, and cross breeding was done among them. Now let's see how gametes will be formed. Now this will be a little different. The gametes will be one capital, one small. In this case, one capital, one small letter. Okay. So once the gametes are formed, they will fuse together. So again we will get 4 different types of combination. First is this combination. It will be both capital T. Second is this kind of combination, capital with small. Third one is this, again capital with small, and fourth is this. Both are small. Okay. So in F1 generation, I told you all the plants were tall. What about F2 progeny? In F2 progeny, 3/4 were tall. Only 1/4 of the plants were dwarf. All these 3 are tall plants, while this is a dwarf plant. Okay. So that means there are 2 types of genes. What are these? Those genes which are able to express themselves, okay, which are visible in F1 progeny, they are called dominant gene. While those which cannot express themselves in the presence of dominant gene are called recessive gene. Okay. So in this case, although one is capital T, another gene is small t, but this small t is not able to express itself, we will not be able to see these characteristics because they are recessive genes. These recessive genes are visible only in the presence of other similar recessive gene. So in this case, both T's are small, so the plant will be dwarf, while all these 3 plants will be tall because capital T is dominant gene. So capital T is present in all 3 of them, so that is why it will be able to express itself. So these plants are tall. So this small t will not be able to express itself because of the presence of dominant gene. but this small t will be able to express itself because capital T is not present in the cell, only two small t's. So, I hope it is clear to you. So, by performing monohybrid cross for F1 and F2 generations, the Mendel, he was able to show that some of the genes are dominant while others are recessive. Now, the next question, how do Mendel's experiment show that traits are inherited independently? So, another kind of experiment performed by Mendel was dihybrid cross, in which he took any two characteristics. So, let us take round and yellow. On the other hand, wrinkled and green. Okay. So, this is capital R means the seeds are round, while Y represents yellow colored seeds. On the other hand, small r represents wrinkled seeds and small y is green seeds. Okay. So, we in this case we are taking into account two different characteristics of the seeds of the pea plant. So, when the cross hybrid was done between these two plants, it was in F1, what was found was all the plants were round and yellow. So, it was expected that all the plants will be round and yellow because it was found that round seeds and the yellow color of the seeds, both these traits are dominant. So, they were able to express themselves in the F1 generation as we studied in monohybrid cross. Similarly, in F1, dominant genes will be able to express themselves. So, all the seeds were found to be round and yellow. But when again cross hybrid was done between these two plants from the F1, what was found in F2? Four kinds of combination. One is round yellow, okay, as expected. Another was wrinkled green. Okay. So, the ratio was 9:1, but a new type of plant was observed in F2 progeny. They were round with green, round with green. Another was wrinkled yellow in the ratio 3:3. So, 9:3:3:1. This was the ratio in F2 progeny when the parents from F1 there was crossbreeding done between these two types of plants. So, that means although we were expecting round yellow and wrinkled green, but this new combination was found in the F2. This shows that the two characteristics, that is either round wrinkled or yellow green, they are inherited separately. Okay. It is not a combination will be inherited. So, round or wrinkled will be inherited separately, yellow or green it will be inherited separately. So, this was shown by Mendel's dihybrid cross. So, let us move on to the next question. So, students, the next question is, a man with blood group A married a woman with blood group O and the daughter has blood group O. Now, which blood group is dominant? Or the question is this information enough to decide which blood group or which trait is dominant and which one is recessive? So, the answer is this information is not enough because both A and O in this case can be dominant. For example, let us assume A is dominant. So, the father can have either, okay, this I with A represent the trait that blood group is A and I with O represents the blood group is O. Okay, so it is O type of gene. Now, since the man has blood group A, so the man can have either pure A genes or he can have this one recessive and one dominant if we consider that blood group A is dominant. And the mother, since she has blood group O, that means mother is has pure recessive genes. So, the chances of combination are 50-50, that the offspring will have this combination or just both oval a combination. You got it? So, in both the cases, I mean, and in these two cases, the child will have, in this case, the child will have O, in this case, the child will have A blood group. So, on the other hand, if we consider that O is dominant, all right, so then in this, if O is dominant, then the father can have this kind of combination. He can have a recessive A. So, in both the cases, it is a possibility that the child will have the blood group O. So, this information is not enough to decide which trait is dominant and which one is recessive. But just for the sake of your knowledge, I am telling you that blood group A and the blood group B, that is IA and IB, both these traits are dominant, while the O gene is recessive gene. Okay. So, if a person has blood group AB, that means his genes contains this A as well as B. He has this type of chromosomes in which both the dominant genes are present and the dominant gene is able to express itself, you know that. So, he has both, that is why the blood group is AB. While on the other hand, if a person has blood group O, then both the traits, they contain O because it is a recessive gene, then there is only one possibility. Okay. Now, let us move on to the next topic. How is sex of the child determined in human beings? So, in human beings, as you know, sexual reproduction takes place. Half of the genes comes from mother and the other half comes from the father. So, a human beings contains 23 pairs of chromosomes. Out of that, 22 chromosomes determine all other characteristics, right from small things like color of the hair, color of the skin, height, functioning of organs and other characteristics, thousands of other characteristics. But the 23rd pair actually determines the sex of the organism. The 23rd pair is different in males and in females. In females, it is X and X chromosomes. This is how we represent it, XX chromosome in females. On the other hand, in males, the 23rd pair is represented as XY chromosome. Okay? So, now first step is gamete formation. That is, the cells, they split into half. This type of cell division is called meiosis, in which the chromosome number decreases to half. Okay. So, the gametes have only X half number of chromosomes. So, out of this 23rd pair, both the gametes in females will be identical because they will have the X chromosome. So, all the ova or the female gametes have X chromosome. But in case of males, what will happen? Half the gametes will have X chromosome while the other half will have the Y chromosome. So, these are 50-50% chances. So, 50% of the sperms formed will have X chromosome, the other 50% will have Y chromosome. Now, during fertilization, when fusion of gametes takes place, what are the chances which sperm will fuse with the ovum? That will decide the sex of the baby. Okay. So, if X fuses with the X, the newborn will be X from the female mother and X from the father. So, it will have XX chromosome and this child will be a girl child. Okay. But if this X fuses with the Y chromosome, so X from the mother but Y from the father, it will be a boy child. Okay. So, the sex of the child depends upon which sperm actually fuses with the ovum during fertilization. I hope it's clear to you. Now, the next question. A study found that children with light colored eyes are likely to have parents with light colored eyes. Now, on this basis, can we say that light colored eye is dominant or recessive? Okay, so as we just discussed in this blood group question also, this information is not enough to decide whether this light colored eye trait is dominant or recessive, because we have to study a large population over generations, then only it is decided whether the given trait is dominant or recessive, because these parents, whether they have pure gene or a combination of dominant recessive, all these things are to be studied. Next question, which type of organism will have more variation, asexually reproducing or sexually reproducing? So as I told you, in asexual reproduction only one parent is involved. Okay, suppose this is an amoeba cell with a nucleus. So what will happen? First, the nucleus will DNA copying takes place and double the number of chromosomes are formed. So in the nucleus, what will happen? Double the number of chromosomes will be formed, and then the cytoplasm will start dividing, and then slowly and slowly this will break into two cells. Okay. So one cell gives rise to two cells. In this case, the chances of variation are very, very low, because the since only during DNA copying only some variation can occur. Otherwise, more or less, the parent cells, they are exact copies of the daughter cells are exact copies of the parent cell. But in case of sexual reproduction, there is a new combination formed. Okay, half chromosomes from the male and half from the female, they fuse together to form a new combination. So that is why these new offsprings are not exact copies of their parents, and there are more chances of variation in sexually reproducing organisms. Next question, what are the functions of chromosomes? So the functions of chromosome, first is during for hereditary. As I told you, in this case also, if you see, this daughter cells will resemble the parent cells because the hereditary material or the genes, they are transferred from the parents to the next generation of springs. Second is sex determination. Chromosomes help in determining the sex of the baby, whether it will be a girl child or the boy child. This is also decided by the chromosomes. And third, one more function of chromosomes can be to bring about variation and evolution. Slight changes in the chromosome in generation after generation gives rise to new species, new kinds of variations in the given species. Next question, why did Mendel choose pea plant for his experiment? As we discussed, Mendel performed various experiments, both monohybrid and dihybrid, to study how genes are actually transferred from parents to the next generations. And for his experiments, he chose pea plant. Why did he do that? Because of various reasons. First reason is that pea plants had certain very distinct characteristics which are very easy to observe. For example, length of the stem. So some of the pea plants are tall while the other pea plants are short. So it is easy to distinguish between tall and the dwarf plant. Then the seeds, some are round, others are wrinkled. The color of seeds is yellow and green. So likewise, there are few other characteristics which are totally different from each other and which are very easy to observe. Second reason is that pea plant is a self-pollinating plant. Okay, so reproduction is easy in these. Third reason is that it grows very fast, so the life cycle is short, so it's easy to perform the experiment in a shorter duration of time. Does genetic combination of mother plays role in determination of the sex? Please refer to question number 9, which I just discussed. I told you how sex of the newborn baby is determined. So mother has both X chromosomes. Okay, in mother XX chromosomes are present, but in males X and Y chromosomes are present. So if X gamete, so in mother all the gametes are similar, they contain the X chromosome. But in father, the sperms are different. 50% of the sperms contain X chromosomes and 50% contains the Y chromosomes. So it is the sex of the newborn baby actually is determined by which sperm first fuses with the ovum during fertilization. So the genetic combination of mother does not play a significant role in determining the sex of the child. It actually depends upon the chromosomes present in the sperm. So that's all from this chapter. I've discussed few important questions along with answers. I hope it's clear to you.